Remind me on Wednesday about the exam format, okay, as far as time goes, because I'd like to try to wrap up everything by this Wednesday so y'all can focus on exams next week, okay? So make sure I talk to y'all about that on Wednesday. All right, we had talked about transcription, and what we're going to move into is the second part of gene expression, which is translation, okay? So for prokaryotes, they don't contain non-membranous organelles, as we talked about before. Both steps, transcription and translation, can overlap for a couple of reasons. One, because there are no non-membranous organelles, and two, even though gene expression in prokaryotes does begin with transcription, the mRNA being synthesized by RNA polymerase in prokaryotes, okay, or the RNA being synthesized by RNA polymerase in prokaryotes, it is mRNA. It's ready to be translated at the ribosome. So therefore, that's what allows these two steps to overlap, is the lack of membranous organelles, and that the RNA synthesized is mRNA. But it still begins with transcription, okay? Then there's a point where they can overlap and it'll end with translation. This is in contrast to eukaryotic cells. Transcription and translation are completely separate for two reasons. The obvious, DNA is stored in the nucleus because eukaryotic cells have membranous organelles. The other reason is that eukaryotic RNA polymerase, when it synthesizes RNA, okay, it's called pre-mRNA. It has to go through RNA processing, such as the addition of a 5' cap, a poly A tail, and what is called splicing. Now, I didn't go over alternative splicing, but make sure you go over that in the recording, the full recording. So once that pre-mRNA has been post-transcriptionally modified to mRNA, then that mRNA is transported out of the nucleus into the cytosol, where ribosomes can now translate it. So this is the part we're going to focus on here. Okay, there are similarities and differences, but I mainly lean towards eukaryotic translation when we go over this, but it's a little bit of both actually. Okay, now translation has more working parts. If you recall with transcription, there's basically one enzyme that catalyzes the process. Now in eukaryotes, we need transcription factors to help RNA polymerase locate a eukaryotic promoter. But outside of that, RNA polymerase catalyzes the three steps of transcription: initiation, elongation, and termination. And the only real help RNA polymerase needs comes in the form of DNA. DNA tech regulatory sequences called promoters and terminators. You know, let RNA polymerase know where to begin transcription and let it know where to end. With translation, there are more working parts. Okay. In total, there are three different types of RNA needed for translation. Okay. The first type of RNA is called mRNA. Now, all three RNAs are single-stranded molecules. Functional RNA, okay, in pro and eukaryotic cells is single-stranded. Okay. mRNA, the M standing for messenger because the message it contains, okay, is a genetic information in the form of codons on how to make a particular polypeptide. Okay, so we need messenger RNA, which we've talked about. And if it's this one here, you can tell by the fact they're telling you this is a cap and this is a tail. This represents eukaryotic mRNA. Already talked about how mRNA contains genetic information on how to make not specifically a protein, but a polypeptide. Okay, and it contains that genetic information in the form of codons. So it's showing you here, we have a gene right here, and it shows you we take a piece of that gene and magnify it, and then they tell you how the reverse strand serves as a template for transcription by RNA polymerase. And RNA polymerase will use this reverse strand DNA template to synthesize mRNA in the forward direction. And all it's doing is making a copy of the genetic information in the form of mRNA to make a polypeptide, which is just a chain of amino acids, peptide bonded together. Said that genetic information on how to make that polypeptide, it comes in the form of codons, which are base triplets. Each codon codes for any one of the 20 amino acids if we're talking about human eukaryotic cells. But I also talked about how some of these codons are redundant, such as for serine. Serine has a total of 6 codons. There's 2 you're responsible for. Tyrosine has several codons. I put 2 that you're responsible for here. But there's redundant. Now I let upper level classes explain why, okay? But you can see in the human cell, there are 20 different amino acids, which are all listed up here, but there's well over 20 different codons that you can find in mRNA. Then I talked about, however, for one of the 20 amino acids, specifically methionine, which goes by the abbreviation MET that I would use on the test, okay, there's only one codon for MET, AUG. And they discovered the reason for this is because this codon AUG, which you find in mRNA, also serves as what we call the start codon. Now, this isn't true for prokaryotic cells. There's a little bit of a difference, so I'm focusing heavily on eukaryotic cells. In eukaryotic cells, though, there might be more than one AUG codon in a single molecule of mRNA. So how's a ribosome going to know which codon is a start codon? Well, it's going to get a little bit of help that I'm not going to go over, okay? But that start codon is going to be towards the 5' end of the mRNA molecule, and that start codon is going to be embedded in a larger sequence called the Cossack sequence. Cossack sequence reads ACC AUGG, and this AUG serves as the start codon. That's what lets the ribosome know, okay, it's over here where I see AUG as part of this larger DNA sequence called the Cossack sequence. That helps the ribosome know I begin translation over here. Now, out of all the codons on this codon table, there are 3 that do not code for any amino acids. These are known as stop codons. You are responsible for any codons I gave you in your notes, okay, which includes these 3 stop codons. You will not find all 3 stop codons in one molecule of mRNA. You will find any one out of the 3 in a molecule of mRNA. Okay, and it's the same for prokaryotes. They have start codons and they have stop codons. For the start codon, it's a little different, okay? But for the stop codons, they have stop codons. Unlike the start codon, which will be located towards the 5' end of the mRNA as part of the Cossack sequence, the stop codons are located towards the 3' end of the mRNA. You will find any one of these 3. Okay, and the codons are UAA, UAG, and UGA. That's the order I always memorized them in. When I took biochem, we were required to know several of these codons. We definitely had to know the 20 amino acids, as well as their 3-letter and single-letter abbreviations and their chemical structures. But for the stop codons, we had to know these 3, and this is the order I memorized them in. First one's UAA. The second one begins with UA but ends in G, and then the third one just flip the A and the G around to GA. There's your 3 stop codons. Okay. Second type of RNA that's needed for translation is tRNA. The T stands for transfer. And what is being transferred, okay, is an amino acid, such as alanine. This is not one of the 7 you had to know, okay, but alanine is one of 20 amino acids in a human cell. What was discovered, okay, is that when a ribosome is performing translation, okay, it gets a lot of assistance from this second type of RNA, tRNA. tRNA, okay, the T not only has to do with, it's responsible for transferring the correct amino acid to the ribosome, okay, but it's a tRNA that also helps the ribosome translate that genetic information in mRNA in the form of codons. Now, mRNA codons themselves are base triplets. Well, what they discovered is that tRNA, or transfer RNA, also has its own type of base triplet, except its base triplet is called an anticodon. So you can see here where they keep repeating anticodon. And that anticodon is going to be depicted as being on the opposite end of where the amino acid would be attached. Listen carefully. mRNA codons are base triplets that code for a specific type of amino acid. Anticodons are base triplets that complement an mRNA codon. What was discovered is that these transfer RNAs will enter a ribosome, but to ensure that the correct tRNA is transferring the correct amino acid, it has to have an anticodon that complements the mRNA codon, okay, so that this base triplet, the anticodon and tRNA, can base pair with the mRNA codon. So if you look at this anticodon and tRNA, it reads AAC. I'm sorry, is that C or G? I think it's G, AAG. Sorry, my eyes are real bad. It's AAG, okay? So this transfer RNA with this anticodon AAG, okay, this anticodon is only going to base pair, all right, with an mRNA codon that reads UUC. And I'll show you this, how the anticodon, this base triplet in tRNA, it helps the tRNA anchor itself, attach itself directly to the mRNA codon. And when that match occurs, that perfect match occurs, that's what ensures the tRNA has transferred the correct amino acid. From there, the ribosome will take that amino acid eventually from the tRNA when it's synthesizing the polypeptide. So the ribosome gets some help there. So here I'm showing you tRNAs have anticodons such as AAG, and if you hear the word codon, I'm talking about mRNA. But in both cases, anticodons and codons, they are base triplets, okay? Except an anticodon is a base triplet that complements any one of these, mRNA codons, except for the stop codons. These three stop codons here, there are no transfer RNAs that have an anticodon that complements a stop codon. So make sure you make a note of that. There are no transfer RNAs that have an anticodon that complements a stop codon. Okay? This is what further ensures that the ribosome knows when to stop translation. It's going to be a different molecule that recognizes the stop codons. But again, you can see here's the anticodon AAG. This anticodon of this tRNA will only base pair with its complement. Okay? Its complement is going to be UUC. So the amino acid this tRNA should be carrying is the amino acid phenylalanine. Okay? So this is how the match is going on. Now, does that mean the ribosome isn't doing anything but taking all the credit? No, it's still doing a whole lot, everybody, a whole heck of a lot, and I'll point that out. The third type of RNA is called ribosomal RNA, or simply rRNA. Now listen, this is way more than what you need for this class, okay? But what I'm trying to show you here is that this is introductory information. You take biochem or an advanced cell and molecular biology class, and they'll tell you, hey, ribosomal RNA, which is shown right here, okay, there are different types. It depends on whether you're talking about prokaryotes or eukaryotes. There are different types of ribosomal RNAs, such as 23S, 5S, 16S in prokaryotes, or these other types in eukaryotes, okay? And they're trying to tell you the function of ribosomal RNA is to pair together with proteins to form ribosomal subunits that will assemble into a ribosome. Okay. Now, do I want you to know all these specific details? No. Okay. What I want you to know is basically mostly what I circled, or what I've circled. For both eukaryotic and prokaryotic ribosomes, they are formed by ribosomal RNA, okay, that complexes or comes together with proteins, forming two types of subunits, either a large ribosomal subunit, or these ribosomal RNA and proteins will form a small ribosomal subunit. And only when a ribosome is going to perform translation, that is the only time the large ribosomal subunit will come together with the small ribosomal subunit to form the fully functional assembled ribosome. That's the only time they come together. In textbook figures, even for advanced classes, they always show the large combined with the small, but that's not true. The only time they come together is when they are performing translation, and we'll go over this in more detail. So what I've circled or written down is what you need to know from this slide. The extra information is just for you in case you end up coming across it in an advanced class. So we need three types of RNA: mRNA, which contains a genetic message, the genetic information from DNA and how to make a polypeptide. And it comes in the form of codons. We need a second type of RNA called tRNA, transfer RNA. It's responsible for transferring the correct amino acid to the ribosome. Well, what ensures that tRNA transfers the correct amino acid? It's embedded in its anticodon, its base triplet. That anticodon of tRNA will complement a specific mRNA codon, not including the stop codons. And then we need a third type of RNA. That's ribosomal RNA, which will come together with proteins to form two ribosomal subunits. A large subunit here in brown and a small subunit here in brown. Now, so by this point, it's like, well, what the heck is a ribosome actually doing? Well, the ribosome is doing this, and the ribosome is doing this. Translation occurs at the ribosome, okay? But it's the ribosome's job to ensure that a specific mRNA codon matches up with the complementary anticodon. So you can see here, here's tRNA with its anticodon matching up with mRNA's codon. That's one of the jobs of the ribosomes. Make sure they match up correctly so the correct amino acid is transferred over. What else is the ribosome doing? It's joining or chemically bonding amino acids together by the formation of peptide bonds. Okay. Well, peptide bonds are covalent bonds. So remember that I talked about that last class. Peptide bonds are covalent bonds. However, here in the black lines, these little black lines, straight or squiggly, each of these black lines represents a peptide bond. So one amino acid will be peptide bond to another amino acid by the ribosome, resulting in the formation of a polypeptide. So since everything is happening at the ribosome for translation, and since the ribosome is performing these two key steps, which are critical, okay, critical, else translation doesn't occur. That's why the ribosome gets credit for synthesis of the polypeptide. So, ribosomes again, this is the second part. We need three types of RNA. One of those RNAs happens to contribute to what else we need for translation, the ribosome, along with proteins. So each ribosomal subunit, the large ribosomal subunit and the small ribosomal subunit, they're made up of ribosomal RNAs and proteins. Now, as I said, these two subunits will only come together when translation is occurring, such as over here. However, the purpose of this here is to show you that when the two subunits complex together, when they come together, they create three sites. And these are the three sites that tRNAs will pass through. And the tRNAs, when they pass through these three sites, they're moving in this direction. But what's actually moving the tRNAs in this direction, it's actually the ribosome which is moving. Okay, so I'll show you in a bit using this side over here. So when the large ribosomal subunit complexes together with the small, we have the creation of three sites: the A site, the P site, and the E site. The A site, well, it's in parentheses what the A stands for, but I like to say the A stands for access. This is the site that a tRNA will access the ribosome. A tRNA will first access the ribosome coming in through here. It'll enter the A site. Okay. From there, okay, the tRNA will then be moved into the P site. Okay. So any tRNA in the A site will then move into the P site. The P here, it tells you in parentheses what it stands for, but what I want you to think of it is as the P stands for polypeptide. It's in the P site that the majority of the time you will find this growing polypeptide or the polypeptide that's being synthesized, okay, attached to a tRNA in the P site. The E site, well, the E stands for exit, so we don't need to change anything. The E site is where the tRNA, after the ribosome has taken its amino acid from it, the E site is the site that a tRNA will exit the ribosome. So a tRNA will enter the A site carrying the correct amino acid. What ensures it has the correct amino acid? Down here, it'll have an anticodon base triplet that complements an mRNA codon. Then from there, It'll eventually move into the P site where now it has the growing polypeptide chain. And then eventually the ribosome will clip that covalent bond linking the polypeptide chain to this tRNA in the P site. It'll take it and hand it over to the one in the A site. And then this one in the P site will move into the E site where it'll exit with no longer having the amino acid attached to it. Okay. So what is actually occurring is the ribosome is moving this way. Therefore, the mRNA and the tRNAs are moving to our left because the ribosome is moving to our right towards the 3' end of the mRNA. It's reading the mRNA from 5' to 3'. Okay? And it's the ribosome that is moving from codon to codon towards the 3' end. That's causing the mRNA to move to our left, and that's helping the tRNAs, okay, that enter the A site, move to the P, move to the E site. It's the ribosome that's doing the moving. As I said before, the ribosome gets credit for making sure the codons are positioned in these 3 sites so that the tRNAs can come in and do their job, and then it's the job of the ribosome to create that peptide bond. Okay. Remember, polypeptides are proteins. Proteins are organic compounds. So you can see here, from here to here, there are a whole lot of covalent bonds present, not including the negative charge. There are a whole lot of single and even double covalent bonds. Only the covalent bond linking one amino acid to the other. That's the only covalent bond that gets a special name, not the rest of them. That's the one that is a peptide bond. I'm sorry, everybody. Now, what scientists discovered is that it's not a 1-to-1 ratio. It's not one ribosome translates one mRNA, resulting in one polypeptide. What scientists discovered is that many ribosomes can translate the same molecule of mRNA into many polypeptides. So down here is an EM. Each of these little roundish rock-looking structures is a ribosome. The thin part here is an mRNA. We call this structure where you have many ribosomes translating that one molecule of mRNA, call it a polysome. Sometimes they'll call it a polyribosome. Now for translation, there are 3 basic steps to translation, just like transcription. Okay, but there are more working parts. There's initiation of translation. This is where the different parts, the mRNAs, the tRNAs, and the ribosomes, because remember, the ribosomal RNAs are part of the ribosomes, so it's where the mRNAs, the tRNAs, and the ribosomes all get into the correct position to initiate translation. Elongation is the second step of translation. Well, just put down next to elongation, this is synthesis of the polypeptide. And then termination. Termination is where we stop translation. Okay, so initiation up here is going to involve the mRNA start codon. I forgot to mention that. And termination down here is going to involve the mRNA stop codon. Okay. It's a cyclical process, the second step, elongation, but it begins, translation begins with initiation. The way initiation works, okay, is that using the 5, if it's eukaryotes, using the 5' cap of mRNA, the mRNA is first attached to the small ribosomal subunit, okay? Or more specifically, the 5' end of the mRNA is attached to the small ribosomal subunit, okay? Second, the first tRNA, okay? The first tRNA, I'm not going to use initiator on the test, but the first tRNA, okay, that has the amino acid met attached to it will bind its anticodon to the mRNA start codon. Okay, so again, the 5' end of mRNA is attached to the small ribosomal subunit. That's first. Second, the first tRNA that has the amino acid met attached to it, okay, will bind its anticodon to the mRNA start codon. So make sure you know these two here. The start codon we talked about, the mRNA start codon is AUG. So what does the tRNA anticodon have to be? UAC. This anticodon UAC is the complement of AUG. What do we mean by that? They can base pair together. But you have to keep in mind in RNA, we don't have the base thymine, we have the base uracil. So for RNA, it's U, not T. U base pairs with A base pairs with U, C base pairs with G. That's what ensures the correct tRNA has entered carrying the correct amino acid. Third, the large ribosomal subunit will attach to the small ribosomal subunit, okay, resulting in the mRNA start codon and first tRNA positioned in the P site of the ribosome. Okay, so next, the large ribosomal subunit will attach to the small ribosomal subunit, resulting in the mRNA start codon and first tRNA positioned in the P site of the ribosome. Overall, this results in the formation of the translation initiation complex. Overall, this results in the formation of the translation initiation complex. Now, is there more going on? Yeah, there's more. Don't worry about this GDP. You get that in advanced classes, okay? That's what I want you to know. Notice the A site is available now. This is the only exception for a tRNA. It's that first tRNA. I'm not going to use initiator on the test. I'll just say first tRNA. This first tRNA will be in the P site. After that, every other tRNA will enter the A site as long as a stop codon does not enter the A site. Second step is elongation, and this is a step that is cyclical. It just repeats itself. Well, you can see here, they show you translation has already begun. So the first 4 codons have been translated. Okay. Now our 5th codon has entered the A site. Over here in the P site, we have our 4th tRNA carrying the 4th amino acid that has been peptide bonded to the other 3 amino acids. This tRNA in the P site, its anticodon is, base paired with the mRNA codon. Notice the mRNA, we have a 5' end on this side, 3' end on that side. Now what happens is the next tRNA will enter. The only tRNA that will be able to stay in the A site, okay, that will be able to stay in the A site, is the tRNA that has an anticodon that complements and binds to the mRNA codon in the A site. If the tRNA entering the A site has the correct anticodon that complements the mRNA codon, down here, they show you the anticodon will chemically bond with the, or base pair with the mRNA codon. Once that occurs, that ensures that this tRNA has just transferred the correct amino acid to the ribosome. Now the ribosome takes over. Okay, so once the correct tRNA has transferred the correct amino acid based on the anticodon complementing the mRNA codon, the ribosome says, okay, I'm going to break this covalent bond holding this polypeptide to this tRNA in the P site, and I'm going to transfer this polypeptide over to this new amino acid down here in the A site. Now notice, our growing polypeptide is now attached to the amino acid that's on this tRNA in the A site. Notice the shape of the amino acid. It's a diamond shape. Diamond shape enters, diamond shapes here, but now it has that polypeptide with 4 amino acids peptide bonded to it here. So now we have 5 amino acids in this growing polypeptide. This tRNA here in the P site, which now has nothing attached to it, the ribosome is going to shift it over to the E site. So now what happens? The ribosome moves, it shifts to the next codon towards the 3' end of the mRNA. It's the ribosome that shifts. Okay? And the ribosome doesn't shift down to the next base in mRNA. The ribosome shifts to the next codon in mRNA. That's this up here. And when the ribosome shifts towards the 3' end, that moves this tRNA with no amino acid to the E site, and now you can see it exits. And then this tRNA here, notice it now is in the P site. If, when the ribosome shifts towards the 3' end to the next codon, if the next codon is not a stop codon, the next tRNA with the correct amino acid will enter the A site, and this process will continue. Eventually, though, the ribosome down here eventually will shift towards the mRNA 3' end to the next codon, and eventually the next codon will be a stop codon. Remember what I said, there are no transfer RNAs with an anticodon that complements and binds to a stop codon. Okay, so that ensures when an mRNA stop codon enters the A site, okay, this ensures that no more amino acids are brought to the ribosome to help start the process of termination. Instead, it's a protein called release factor that often goes by the abbreviation RF, or if it's in eukaryotes, they call it ERF. I'll just call it RF for the test. In prokaryotes, it's just called RF. In eukaryotes, it's called ERF. Okay. Release factor here. This is the protein that is the only one that has the ability to recognize any of the 3 stop codons. A single molecule of mRNA towards its 3' end will have any one of these 3 stop codons. Not all 3, but any one. When that stop codon enters the A site, there are no tRNAs that have an anticodon that recognizes it. Doesn't matter. Release factor has been waiting around the whole time for a stop codon to enter the A site. When an mRNA stop codon enters the A site, release factor will enter the A site, bind to the stop codon, triggering the ribosome to release the polypeptide. It says free, but normally my professors would say release the polypeptide. Second, it will trigger the disassembly of the initiation complex. It will cause a large ribosome to release from the small, thereby releasing the mRNA. So again, when a stop codon enters the A site, release factor will enter the A site, bind to the stop codon, triggering the release of the polypeptide, and then the disassembly of the initiation complex. Thus, translation is terminated. That's all I'm going to cover for this, okay, everybody? But if everything goes well, by the time translation is completed, we have synthesis of a polypeptide, a long chain of amino acids that represents the first of 4 levels of protein structure, the primary structure, which is simply not only the chain of amino acids, but the order or the sequence of amino acids, such as glycine, proline, threonine, glycine, threonine, all the way down here to the tail, the carboxyl tail, proline, lysine, and glutamate. Okay, so that's what we have. That's as far as I'm going to go here. Are there any questions on that before I jump over to 18? Okay, listen carefully. With 18, I'm only going it covers 2 parts. Okay, but first of all, 18 has to do with regulation of gene expression. Synonym for regulation is just simply control. So we're talking about control of gene expression. Now, I discussed both prokaryotes and eukaryotes. I believe I begin with prokaryotes, though. So listen carefully. I'm going to leave eukaryotes to you to go over. I'm going to focus on prokaryotes, okay? There's something that's unique to what they discovered with prokaryotes, specifically bacteria such as E. coli, that they haven't discovered in any eukaryotic living organisms. Doesn't matter if they're unicellular or multicellular. Okay. In chapter 17, I do talk about that when pre-mRNA is processed, it goes, one of the three modifications it goes through is splicing. What I didn't go over was alternative splicing. Okay. That's where one pre-mRNA molecule, okay, not only has introns that don't code for any polypeptide, not only has introns that have to be removed, okay, but sometimes what will happen is what we call alternative splicing. For eukaryotic pre-mRNA, when it undergoes alternative splicing, not only are introns removed, but some of the exons that do code for the polypeptide may be removed. Scientists discovered the purpose for this is so that the genetic information stored in one gene, okay, when that gene is transcribed in the pre-mRNA, that pre-mRNA can actually be spliced alternatively into two or more different types of mRNA that code for two or more different types of polypeptides, thus leading to two or more different types of active proteins. In prokaryotes, they found out something different. Prokaryote cells or organisms seem to have evolved to where if there are two or more proteins that a prokaryotic cell needs to perform the same cellular function, apparently at the level of the gene, you will find those genes that code for those two or more proteins that perform the same cell function. You will find those two or more genes side by side, okay, and downstream of one promoter. And this is what we're going to get into. But in either case, it's still going to have to do with regulation of gene expression control. Okay, so this chapter is going to focus on regulation of gene expression at the level of transcription. The first part of the notes go over prokaryotic cell gene regulation, and then the second part of the notes will go over eukaryote. I'm going to focus here on prokaryote. But remember, both parts focus not only on gene regulation, the control of gene expression, in other words, the control of when a polypeptide is produced, but both sections focus on regulation at the level of transcription. Will transcription be allowed to occur or not? And if it does, how much? In eukaryotic cells, you can see here, they're showing you, this is an example of a eukaryotic cell. What they're showing you here are these pipes, okay, that imagine water flows through. And instead of showing you switches that look like on-off switches, like light switches, notice they show you these valves, comparable to what you might see outside if you have a house or if you're at your parents or wherever, and you have those valves connected to the pipes to allow water to flow through the hose or out of the water faucet. Okay. The reason why they don't use light switches and instead they use valves is because when it comes to regulation of gene expression, what they're referring to is for some genes, the expression of those genes is like a light switch, on or off. The gene expression of the gene is either occurring, it's turned on, or expression of the gene is turned off. So the cell is either producing polypeptide or it's not. But for many genes, it was discovered it wasn't an issue of turning on and off gene expression. Okay, it was a matter of when gene expression is turned on, how on is it? Such as taking one of these valves at any level of gene expression in a eukaryote, and when you turn that valve clockwise, water, say, will start to flow through the pipe. The more you turn it to the right, the faster and the more water will flow through the pipe. But if you start turning it back to the left, that slows down the flow of water as well as the amount of water going to the pipe. Thus, gene expression, sometimes it's on, but it's on at a very low level, therefore very little polypeptides being produced. But then a cell receives a chemical message in the form of a ligand telling that cell, hey, look, that gene that you are expressing at a very low level, I need you to enhance it, increase that level of expression so we get even more polypeptide being produced quicker. Or a cell may have a gene being expressed at a high level, okay, where a lot of polypeptides being produced, but then the cell will receive a signal in the form of a ligand telling it, hey, look, I don't want you to shut down expression of that gene, but I want you to inhibit it. I want you to suppress or repress it, slow it down. So I want you to turn down gene expression to where now we're getting less polypeptide. So it's anywhere between gene expression is turned on or off, or it's increased or decreased. Okay, so it's not that simple, just basically. Okay, listen carefully. For this chapter, you first have to start off by knowing the basics of transcription. First of all, you need to know for transcription to occur, what type of template do we need? A DNA template. What's going to be produced at the end of transcription? You need to know it's RNA. And you also need to know a third key point about transcription. What catalyzes transcription? What makes transcription happen? RNA polymerase. I can't emphasize enough, if you don't know these three basics going into chapter 18, it's a disaster. Okay? So those are the three key points at a minimum you need to know about transcription. We need a DNA template to synthesize RNA. What is actually using DNA as a template to synthesize RNA? It is RNA polymerase. Then you want to build on that. Okay? For the DNA template used by RNA polymerase, is it both strands or one? No, it's only one. It's only the reverse strand that's used as a template. And the RNA that is made, well, the RNA could be mRNA that's produced by RNA polymerase, or it may be called pre-mRNA. Well, listen carefully. What I'm going to go over is just the section having to do with prokaryotes. Pre-mRNA is synthesized by RNA polymerase in eukaryotes. Prokaryotic RNA polymerase, when it synthesizes RNA, it is mRNA. That's why, even though in prokaryotes, gene expression does begin with transcription, because prokaryotic RNA polymerase is synthesizing an RNA that is called mRNA, that is ready to be translated, this is what allows, at some point, transcription and translation to happen in prokaryotes. But it'll begin with transcription and with translation. But since prokaryotic RNA polymerase is synthesizing not just RNA, but mRNA, okay, that's ready to be translated, the two steps can overlap. Well, why is that important? Because I said I'm only going over the first part of your notes. This is important to remember. But it all starts with these three key points. We need a DNA template to make RNA during transcription, and RNA polymerase synthesizes it. Okay. Now, the other information we need, okay, on top of that, is that in order for RNA polymerase to locate a gene, it needs to find a DNA regulatory sequence called a promoter. The promoter is always upstream of a gene. It says transcription unit here, but what they mean here is gene. Okay. What they're not, and that's here, and it's the DNA. It's highlighted in green. What they're not showing you on this end, what is missing on this end, everybody? What's missing on this end that has to do with DNA? It's a regulatory sequence, but what do we call it? I say it's always downstream of the gene. What are they not showing you here? What's the other sequence? It tells RNA polymerase when to stop transcription, when to stop making the RNA. What do we call that other DNA sequence? Nope. The third step of transcription gives you a hint of what the name of that sequence is. It's also in your lab. Y'all haven't started lab 9 day 1? Because it's in that lab too. It's called a Terminator sequence. All right. Termination or Terminator sequence. Okay. Promoter and Terminator sequence is referring to DNA regulatory sequences. One helps RNA polymerase locate the beginning of a gene. The other one tells RNA polymerase when to stop transcription. You're going to hear talk about these two. Okay, listen carefully. When it comes to prokaryotic gene regulation, scientists discovered that in prokaryotes, specifically bacteria such as E. coli, they tend to have their genes clustered together. And what I mean by clustered together, they tend to have their genes grouped together in a group. And that's what they're showing you here. They're showing you a total of 5 genes that are clustered or grouped together. And what scientists discovered, okay, is that in prokaryotes, such as the bacterium E. coli, where you find these cluster or group of genes side by side, one after the other, grouped together, they also found that they're regulated or they're downstream of a single promoter. So the way they say it is regulation of these, expression of these genes at the level of DNA is under the control of a single promoter. Now this is the problem, okay, what I run into. I don't have students understanding how serious they need to be when they study and how efficient they need to be. Okay, quantity of time isn't going to cut it if your time isn't, if the quality of that time isn't there. Okay, notice these areas that I boxed in that are highlighted in different colors, they're referring to this molecule here. So you have to know when I'm referring to DNA versus when I'm referring to a protein enzyme such as RNA polymerase. And I do have students that understand the difference, but I have a lot of students that don't. They don't even know where to apply the terms, okay? So you have to have those down, please. So what scientists discovered is that in some prokaryotes, such as the bacterium E. coli, you can find a cluster or a group of genes side by side, okay, downstream of a single promoter, okay? When you find that arrangement, they refer to that arrangement as an operon. And I have it down here. An operon is the entire stretch of DNA that includes the operator, the promoter, and the genes they control. It also contains a terminator sequence, which they're not showing you here. Okay? So on this figure, here's the promoter. Here's the group or cluster of genes. Okay? I will talk about the operator later, which is this stretch here. I'll explain what that's about. Okay? Now, this overall structure consisting of a promoter, an operator, and the group or cluster of genes, this whole structure makes up an operon. Now, the purpose of the operon in prokaryotes such as the bacterium E. coli, well, it had to do with this. Okay? The genes that you find grouped together, those genes will code for 2 or more proteins, okay, that all take part or perform the same cellular function, such as the metabolism of lactose. Lactose is a disaccharide made up of 2 monosaccharides. Prokaryotes such as the bacterium E. coli will take lactose, break it down into its monosaccharides, and use those monosaccharides to produce energy or to transform into other compounds. Okay, so lactose is a nutrient that needs to be metabolized or broken down. Well, scientists discovered there's a group of genes, a cluster, okay, side by side, under the control of one promoter and one operator. All right, and all those genes code for proteins that are enzymes that all perform the same cellular function, lactose metabolism. So this is what we're going to go over in a sec. Okay. So these operons consisting of a promoter, an operator, and say 3 or more genes side by side, okay, they code for proteins that perform the same function. Well, they found these operons can be divided into 1 or 2 groups based on when they're expressed or how they're expressed. Okay, so there are operons referred to as inducible operons. Okay, inducible operons are normally turned off. That means gene expression is not occurring. No protein is being produced. Okay, but the reason why they call these inducible operons is because something has to occur to where they're induced or to where gene expression is induced to turn on. Okay. An example of an inducible operon, a specific example of an inducible operon that we're going to go over, it's called the Lac Operon. Okay, so there are operons referred to as inducible operons. Normally, gene expression of these operons is turned off. There has to be something that induces gene expression of the operon. In other words, it causes gene expression to be turned on. An example of an inducible operon we're going to go over is called the Lac Operon. The lac here is in reference to lactose. So when we go over this Lac Operon, we're going to talk about how this operon consists of a group of genes that metabolize lactose, break down lactose. But we're also going to talk about how lactose is responsible for inducing gene expression, for turning it on. So it has a dual function. Another type of operon, or there are other operons, I should say. There are other operons whereby gene expression is normally turned on. So these genes for these operons are normally being expressed. Protein is being produced, and the same cellular function is occurring. Okay, and there has to be some event, okay, that represses gene expression. Okay, that turns off gene expression. We call these operons repressible operons. Okay, so in the name of these two groups of operons is what has to occur to change their state. Change them from an inactive state to an active state, or from an active state to an inactive state. That's what we're getting at. So the type of operon we're going to go over here specifically, an example of a repressible operon, is the Trp operon. Okay. Trp stands for the amino acid tryptophan. And we're going to talk about how this amino acid tryptophan, how bacteria, prokaryotes such as the bacterium E. coli, they have the ability to synthesize their own tryptophan if it's not in the nutrients they absorb. But because they have that ability to do that, they also need to make sure that they don't have too much tryptophan. So we'll talk about how where E. coli are synthesizing tryptophan, but then if it's suddenly in the environment around them that they can absorb it as nutrients, okay, this same amino acid tryptophan that the bacterium is synthesizing, this amino acid will also play a role in repressing gene expression, turning it off. So it'll also have a dual function, just like lactose. Okay, so this is a Lac Operon. Listen carefully. There's a lot more detail I want you to know for the Lac Operon than for the Trp operon, okay? As far as names of genes and names of protein enzymes, okay? For the Trp, there's still a lot, and it's still hard, but you won't have to know the specific names of genes. You'll just have to know how many genes. And from that, you'll have to know, based on how many genes, how many active proteins end up being produced when those 5 genes are being expressed, because it's different between the two operons. It's not just the difference in the whether they're in an active state or inactive, or vice versa. Okay, it has to do with the number of genes that make up the operon. There's an operon called the Lac Operon. The Lac, again, has to do with lactose. Okay, this Lac Operon consists of a promoter, which is a DNA regulatory sequence, an operator, which is also a DNA regulatory sequence, okay, and 3 genes that code for 3 polypeptides, and each polypeptide folds into a tertiary structure, becoming an active protein enzyme. These genes, in the order that they're transcribed, are LAC-Z, LAC-Y, and LAC-A. Okay? The LAC-Z gene codes for a single polypeptide that folds into an active protein enzyme known as beta-galactosidase. This gene was used quite a bit in labs, and in labs, whenever the protein enzyme was produced using this gene, instead of saying beta-galactosidase, they would say beta-gal, so know both. Okay? There's the second gene called the Lac Y gene. This gene codes for a single polypeptide that folds into an active protein enzyme known as permease. And then there's a third gene that makes up this operon, and this is the third and final gene. It's called the Lac A gene. It codes for a single polypeptide that folds into an active protein enzyme called transacetylase. So all three of these protein enzymes, active protein enzymes, they exist at the tertiary level of protein structure. So Lac Z gene codes for beta-galactosidase, Lac Y, permease, Lac A, transacetylase. All three end in ASE. All three are involved in the same cellular function. Okay? All of them are involved in the metabolism of lactose. Okay? Beta-galactosidase takes the disaccharide lactose, okay, breaks the covalent bond holding the two monosaccharides together that form lactose, galactose and glucose. So beta-galactosidase acts on the substrate lactose, breaks it down into two products, the monosaccharides galactose and glucose. Permease's job. When E. coli, when prokaryotes such as the bacterium E. coli are in the presence of lactose, some of that lactose is absorbed, okay, resulting in the activation of the Lac Operon. And once permease is produced, permease will increase the amount of lactose that is absorbed by the prokaryotes such as the bacterium E. coli. So that's its job. Lactose absorption, beta-galactosidase, the breakdown of lactose, transacetylase, function unknown. I haven't looked up its function in about 5 years and it got ugly. It went from being unknown to being known, but nobody wanted to say what they knew because it wasn't easy. These two enzymes, very clear-cut functions, okay? This one, just leave it as unknown because it's ugly, what I came across so far, all right? But transacetylase, along with beta-galactosidase and permease, all three of these enzymes performing cellular function, which is lactose metabolism, okay? But notice something. I'm telling you down here also that this disaccharide lactose is not just a nutrient that prokaryotes such as bacterium want to metabolize, okay? It also functions as an inducer. Lactose induces gene expression of the Lac Operon, okay? So let me go back to this figure here. The way it works, okay, is RNA polymerase, okay, needs a promoter, and it'll locate the promoter of the Lac Operon. This promoter will be upstream of a group of 3 genes, these 3 genes being Lac Z, Lac Y, Lac A. And when possible, RNA polymerase will bind to the promoter, which helps it locate the Lac Z gene, where it will initiate transcription. But the genetic information for these 3 polypeptides that form 3 different enzymes, okay, the genetic information in all 3 genes will be transcribed into a single molecule of mRNA. Except notice something. In the mRNA, they're showing you a green bar, a red bar, a green bar, a red bar, a green bar. What do you think those bars stand for, everybody? What do you think those green and red bars stand for in the mRNA? Any ideas? I talked about it today in chapter 17. Not promoter and termination. We're talking in mRNA. So you're right, Rice. Start and stop codons. Okay. Remember, remember, if it's promoter or termination, or terminator, we actually say terminator, not termination. If it's a promoter or terminator sequence, we're talking about DNA. Okay. But this is an mRNA. I'm asking you what the green and the red stands for in the mRNA, and it's start and stop codons, okay? Yeah, and it happens. So, but this is why I'm asking you questions. I'm not, listen, something I should have clarified a long time ago, okay? Even if you give me a wrong answer, as long as it has to do with the topic recovery, sometimes, I don't know about y'all, but I hate to say it, but I've learned a lot more from mistakes than when I'm correct. So it's good to do this, okay? If you hear the terms promoter, terminator, or now operator, think DNA. Okay, but if it's in mRNA, we talk about start and stop codons. So even though all three genes are transcribed as a single mRNA molecule that is immediately ready for translation, because remember, this is prokaryotes, mRNA is synthesized. The ribosome, when it translates this one molecule of mRNA, that ribosome recognizes, oh, this is one molecule of mRNA, but it contains three different pieces of genetic information. Right, exactly. So what'll happen is the ribosome will find this first start codon and start to translate it until it reaches this first stop codon. That lets it stop making the polypeptide, which becomes beta-galactosidase. But right when it's going to let go of the mRNA, because it encountered this first stop codon, right when it's going to let go of the mRNA, it realizes, oh, there's another start codon. Hold on, everybody. We're not going to disassemble yet. We're not going to terminate. Let's restart, okay, the initiation, or restart translation at this second start codon to the second stop codon. Second polypeptide's made that becomes permease, and right when the ribosome wants to terminate translation, all of a sudden it sees another start codon and says, wait, we need to continue with translation. Finally, when it reaches that third stop codon of the Lac A gene here, that came from the Lac A gene, when it reaches that third stop codon and synthesizes that third polypeptide and becomes transacetylase, that's when translation will be terminated. Except for the Lac Operons, the reason why they're used as an example of gene regulation is because every operon comes with an additional sequence called an operator. And the operator is recognized and bound by a repressor protein. The repressor has to do with it's an inhibitor. Okay, so as the promoter, you can think of it serves more as an activator. It helps in turning on gene expression. The operator acts more like a repressor or a silencer, or simply an inhibitor. The operator's role is to inhibit the expression of the Lac Operon genes. It's to inhibit RNA polymerase. So notice this DNA regulatory sequence called the operator, it is located in between the promoter and the first of three genes of the Lac Operon. This operator, this DNA regulatory sequence called the operator, okay, it can be bound by a repressor protein. If the operon is the Lac Operon, we simply call the repressor protein the Lac repressor. They show you what I told you all in the past. A protein's shape is directly related to a protein's function. So what was discovered is that this repressor protein, okay, has two binding sites. The binding site on top here, if you notice, they give it a shape that complements the shape of the operator, indicating that this Lac repressor protein has the ability to recognize and bind to the operator. Well, notice where the operator is situated, in between the promoter and Lac Z. So whenever the Lac repressor binds to this operator sequence, it's able to inhibit RNA polymerase by physically preventing it from sliding over to the first gene. But I said it has two binding sites. This second binding site, I'll show you in a sec more clearly, where I have an arrow pointing. This second binding site is for the sugar lactose, which I'm just going to write as LAC. And the way it works for this Lac repressor is that even though it has two binding sites, the one on top for the operator and the one on the bottom for the disaccharide lactose, both binding sites do not work at the same time. What scientists discovered is that if the Lac repressor has a choice at the same time of either binding to the operator or to the sugar lactose, it prefers lactose if it has the option, but it can't bind to both. Therefore, the way we go over this, the Lac operon is an inducible operon. Therefore, gene expression is usually in an off state. That's what they mean here. By saying operon's off, gene expression is off. So the Lac operon is an inducible operon. Normally, gene expression is turned off. Why is it turned off? Because the Lac repressor protein here, which is coded for by a different gene that I do want you to know, the Lac I gene. The LAC-I gene is a different gene. It is not part of the Lac Operon. It's a separate gene with its own promoter. But this LAC-I gene codes for the Lac repressor protein. When lactose is absent, this Lac repressor protein is in an active state. Okay? Its binding site that recognizes the operator, that binding site of the Lac repressor protein, okay, has a shape that complements the shape of the operator. Thus, the Lac repressor protein is active, and it will use this binding site to bind to the operator of the Lac Operon. Again, that operator is situated in between the promoter of the Lac Operon and the first of 3 genes of the Lac Operon. Therefore, when it binds to the operator, it physically inhibits or prevents RNA polymerase from being able to slide over to the first gene. Thus, gene expression of the Lac Operon is in an off state. Okay? Down here I mentioned the 2 binding sites again. But what happens when lactose is present? Well, as before, actually let me do it this way. What happens when lactose is present? Well, as I said before, lactose is actually the inducer. So the Lac Operon is an example of an inducible operon. Inducible is referring to a molecule that acts as an inducer, causing the expression of the causing gene expression to be turned on. So lactose, when lactose is present, okay, and the Lac repressor has a choice, the active Lac repressor has a choice between binding to the operator or binding to lactose. It was discovered that the Lac repressor protein prefers to bind to lactose. However, when the Lac repressor protein binds to lactose, that causes the Lac repressor protein to change its shape. This results in the other binding site that when the repressor is active, complements the shape of the operator. This results in that other binding site no longer having a shape that complements the shape of the operator. Thus, when the Lac repressor protein binds to lactose, that change in shape that's caused by the binding to lactose, that inactivates the Lac repressor protein. That other binding site doesn't have a shape that complements the operator. Thus, we say the Lac repressor protein has now been inactivated. It cannot bind to the operator. RNA polymerase, which has been right here by the promoter the whole time, finally says, okay, this guy's out of my way. Therefore, I'm not being inhibited, okay, from sliding over and turning on gene expression of the Lac Operon by first initiating transcription. So when we talk about operons and gene regulation, okay, of the operons, it's at the level of transcription that we're talking about gene regulation. Okay? Well, there's another type of operon, as I said before. This other type of operon is known as a repressible operon. Okay? These operons, gene expression is normally turned on. The specific example I want you to know of a repressible operon is a trip operon. Okay? Uppercase T, RP represents the amino acid tryptophan. Outside of that, we use a lowercase t when we're talking about the operon. Okay. Listen carefully. You do not need to know the names of the genes or the enzymes. You just need to know the number. So first of all, the trip operon consists of 5 genes that code for 5 polypeptides. These 2 genes are the polypeptides they code for form 1 functional protein enzyme. These 2 genes down here, they also code for 2 separate polypeptides, but those 2 polypeptides form a second functional or third type of protein enzyme. And then this 5th gene here, it codes for a polypeptide that becomes a functional enzyme. So total, the trip operon consists of 5 genes that code for 5 polypeptides that form 3 functional protein enzymes. So that's the key I want you to know. This is another figure showing it to you. Here are the 5 genes that code for 5 polypeptides. Each of these polypeptides will fold into a tertiary structure called a protein subunit. And then, as I said, then what will happen is, 4 of those polypeptides will form 2 enzymes total. Now listen carefully. The way it works here is a trip operon. Gene expression is normally on. Okay, 1 mRNA is formed using these 5 genes, translated into 5 polypeptides that form 3 protein enzymes. These 3 protein enzymes, okay, working as a group, and they catalyze a series of chemical reactions beginning with a precursor that eventually results in the production of tryptophan. Gene expression of this operon is on unless tryptophan is provided in the surrounding environment. If suddenly tryptophan becomes available in the surrounding environment, in the nutrients being absorbed by a prokaryote such as the bacterium E. coli, now what happens is within the bacterium such as E. coli, tryptophan levels, okay, become too high. There's more tryptophan present, okay, than the bacterium needs to make proteins or make polypeptides. So what happens is any excess tryptophan, all right, will negatively feed back on gene expression. In other words, any excess tryptophan will function as a co-repressor, having a negative effect on gene expression of the trip operon. What will happen is that excess tryptophan will turn off gene expression, okay, of the trip operon. This ensures that prokaryotes, such as the bacterium E. coli, don't have too much tryptophan or too little. To prevent too little tryptophan from being present, operon gene expression is on. So there's always just enough tryptophan. But should suddenly excess tryptophan become available in the diet of the bacterium to where now there's too much tryptophan, not a problem. That same product being produced by the genes that make up this operon, well, that same product is also going to act as a co-repressor, turning off expression of the genes that produce it. It's kind of a auto-regulatory process. Let the product help control when expression of this operon is occurring in order to maintain the ideal amount of tryptophan or concentration of tryptophan in the cell. So the way it works here, okay, again, it's an operon. It consists of a promoter, an operator, and a group of genes. In this case, 5 genes. You don't need to know the specific names, just know it's 5 genes. Okay? And in this case, though, since the trip operon is a repressible operon, that means normally gene expression of the trip operon is turned on. When it's turned on because there is not excess tryptophan available. So I'm going to put excess. Doesn't mean there isn't any tryptophan. It's just there's no excess tryptophan. Whatever tryptophan is present, it's being used by the cell to make polypeptides. So there's no excess tryptophan. So when there's no excess tryptophan, okay, there's a repressor protein. And I said the name of the repressor protein comes from the name of the operon. So if it's a Lac Operon, the repressor protein is called the Lac repressor protein. If it's a trip operon, you can see down here we have our trip repressor protein. Now the trip repressor protein also has 2 binding sites. Okay, one of the binding sites is for the operator, the other is for the co-repressor tryptophan, any excess tryptophan. What was discovered, okay, is when there is no excess tryptophan or tryptophan is absent, okay, the trip repressor protein is in an inactive state. The shape of the trip repressor protein, okay, on the side where it binds to the operator, that shape of the trip repressor protein doesn't complement the operator, so it can't bind. Okay? Therefore, under normal circumstances, since the trip repressor protein is inactive, gene expression of the trip operon is on. RNA polymerase can freely bind to the promoters, slide over to the first of 5 genes, in the Tryptophan transcribe all 5 genes into one molecule of mRNA, and then the ribosome will recognize there are 5 start codons and 5 stop codons. Therefore, it knows to synthesize 5 separate polypeptides. Again, these 5 separate polypeptides will end up forming a total of 3 functional protein enzymes that will synthesize tryptophan, as it says here. That's when there's no tryptophan present, or what they actually mean, not excess. However, when tryptophan is present, okay, well, the Tryptophans are repressible operon. Therefore, when tryptophan is present, in other words, when there's excess tryptophan or too much, this results in the repression of gene expression. What do I mean? This results in gene expression of the Tryptophan being turned off. Well, what was discovered is that excess tryptophan that's not being used for the synthesis of polypeptides, that excess tryptophan will bind to one of the 2 binding sites of the repressor protein. And when tryptophan binds to one of those 2 binding sites of the repressor protein, it causes that repressor protein to change its shape. Okay, so I'm just going to put this here, shape. I should have did that in the other one, but we're running out of time. So it'll cause that repressor protein to change its shape. So when tryptophan binds to one of the 2 binding sites on the repressor protein, the repressor protein will change its shape, becoming active. What do we mean becoming active? Now the other binding site has a shape that complements the shape of the operator, thus allowing the repressor protein to bind to the operator. And again, you can see here, and again, when it binds to the operator, it's in between the promoter and the first gene, the first of 5 genes in the Tryptophan, thus physically inhibiting or repressing gene expression. In other words, turning off gene expression of the Tryptophan by physically blocking it. Now please notice something. Unlike the Lac repressor protein, the repressor protein, okay, both binding sites can work and do work at the same time. This is not true for the Lac repressor protein. Lac repressor protein is either binding to the Lac operator or it's binding to lactose. Can't bind to both. But for the repressor protein, both binding sites that I've listed down here, both binding sites do work at the same time when this both to activate the repressor protein, and so the repressor protein actually inhibits or represses gene expression. Okay. Are there any questions on this trip one? Please remember, I know they give you the genes here, okay, but you don't need to know the names of the genes. Just know there's 5 genes in the Tryptophan that code for 5 polypeptides, and those 5 polypeptides form a total of 3 functional protein enzymes that are all involved in the same function, okay? Tryptophan synthesis, okay? And where it says tryptophan is absent or tryptophan is present, what they're referring to is tryptophan is present. They're basically referring to there's an excess, but going into the test, you can think of it as present or absent. But technically speaking, they're referring to, is there too much tryptophan present or is there not enough present? 5 genes, 5 polypeptides, and 3, enzymes that all synthesize tryptophan. And then other than that, Mac, just know how the operon works, right? Tryptophans are repressible operons, so you have to know that means, okay, that gene expression is on. That's where you start. And then you work your way towards gene expression being turned off. It's the opposite for the Lac repressor. Tryptophan is one of 20 amino acids. So I put that up on here. I did, right? Oh, I thought I did. Oh, I'm sorry. Wait a minute. Yeah, I think I have it in your notes if I didn't have it here. I thought I had it on here. Okay. I do mention it down here though. Okay, but tryptophan is an amino acid. So in humans, it's one of 20 amino acids. I don't know how many amino acids total are in the bacterium E. coli because most of this information comes from E. coli. Okay, but if you apply a tryptophan is an amino acid that you find in humans, that you find in E. coli, and most of not all living organisms. It's just a matter of the total number if there's any difference. Okay. Yeah, that's why it's important. Okay, everybody. Again, don't let me forget on Wednesday to talk to you about the time format for the exam. Make sure you or listen to the rest of the recording for chapter 18. I'll cover chapter 19, and if there's any time left at the end, you can ask me any additional questions you have on the chapters we covered, um, or just let me know if you need to meet with me, or just send me an email. Okay. Okay, everybody.